nervous system development, and I'll continue talking about the vertebrate nervous system development. And before I go on to today's topic of division and temporal specification or timing, I'll just like to briefly recap or recapitulate what we discussed last time so that you can briefly go over it, and then just we'll headlong into the topics of discussion for today. Okay. This is the overview of the entire class, which Sonia has already covered the invertebrate part of the nervous system development, and I'm talking to you about the vertebrate nervous system. So we started, both of us, by saying that how a neurogenic region is specified in the developing embryo, and some of this is conserved between across evolution. In case of the vertebrate, there is this neural plate and the formation of the neural tube, followed by spatial patterning, which is across several axes, the anterior, posterior, the dorsoventral. And today in my talk, I'll be talking cell division and the temporal patterning. Right. So just very quickly to go over some of the slides. So this is the neural tube patterning. As you can see, the neural tube is closed here. Some of the mesodome goes on to form the somites, which form the musculoskeletal system. Then this is the top view. And as you can see that, the very midline forms the neural tube. And this is in fact, the anterior posterior axis is governed by BMP signaling, where very anterior, the inhibition of BMP leads to the formation of the neural ectoderm. And in the dorsoventral axis, it's the sonic hedgehog and the BMPs, which pattern the entire dorsoventral axis, particularly at the level of the spinal cord, where all the different zones and the different neurons are specified with the sonic hedgehog from the bottom and the BMPs and the winds from the top. And I also talked to you about the signaling center in the developing forebrain. The forebrain goes on to form your cerebral cortex, which is the seat of all higher order functions. And how I told you how an FGF8 signaling at the very, very rostral or the nose end patterns the entire cortex, like forms the frontal, decides where your frontal, parietal, and the occipital lobes will form. And the frontal cortex is where your motor cortex comes, which is involved in movement. Parietal is for touch, somatosensory cortex, and the occipital lobe is the vision, right? And I also told you the same, how FGFs play a role in making the dorsoventral patterning as well in the forebrain, right? Now, today in my talk, I'll be, as I told you, we'll be covering about divisions. So I'll start with telling you about some of the features of the nervous system, particularly the vertebrate nervous system, and how it is evolved to be more complex than simple organisms, organisms which don't have such subdivisions. And anywhere, feel free to stop me and ask questions. Probably what I'll do is at several different checkpoints, stop and check up on you, whether you're all on board with me today, right? So a feature of this mammalian nervous system is that the nervous system consists of the central nervous system and the peripheral nervous system. And the central nervous system is, just hide this, okay. The central nervous system consists of the brain and the spinal cord, whereas the peripheral nervous system is the somatic and the autonomic nervous system, which will become very clear in this next slide. And the nervous system is in the all total nerve connections, everything which is there in your body. And something which will become clearer to you once you look at the schematic is the CNS is all that is peach colored here. So basically the brain and the spinal cord is all the central nervous system, whereas the peripheral nervous system is all that nerves which go from the brain and the spinal cord to the body and come back from the body to the brain and the spinal cord. And this consists of two, the parasympathetic or the sympathetic, which are the somatic and the autonomic nervous system. Basically, these mostly control all your, the autonomic controls all your involuntary mechanisms, like, your breathing, heart rate, peristaltic activity, and all of that. So this is the sum total of the entire nervous system. And as I've been telling you before, what I've really focused on is the development of the central nervous system, particularly looking at how the brain and the spinal cord develops. And these peripheral nervous system all comes from the neural crest cells, which, if I can go back, comes from the neural crest cells, which are, though not shown here, the neural crest cells give rise to the peripheral nervous system, right? Okay. And just to, today I'm going to show you a lot of sections. So just so that you orient yourself to how a brain is cut, I've shown the different directions in the nervous system and the planes at which they are cut. So a coronal section or a coronal place, it's in the middle. It's basically when you cut face-wise, and that's called a coronal plane. And if you cut it face-wise, you will see a brain which looks like this. Is from the back of the brain. You can see the spinal cord, you can see the cerebral cortex as well, and you can see different other structures in the brain. Now, a sagittal plane would be a sideways cut. And as you can see here, this gives you the entire view of the brain. You can see the nose here, you can see the eye socket, you can see the entire cerebral cortex where my arrow is moving. And you can see the cerebellum, and you can see the medulla oblongata going into the spinal cord. And a horizontal plane, which perhaps is not important today, but just it's a plane across, you know, when you would cut parallel to the ground, and this is how the brain will look from the top, right? So the structure, and this is a human brain. The structure of the brain is that this is a whole brain, and if you would cut a coronal, a sagittal view, you will see that the brain has these different many structures. And some of the important things which I would like to highlight for my talk today is that this is the cerebrum, and there are different terms which are used for the cerebrum. It's called the cerebral cortex. It is also, the cerebral cortex consists of the neocortex, which has the very six-layered structure. It also consists of the hippocampus. Together they make the cerebral cortex. So this is the cerebral cortex. This region here is the hypothalamus. This is the pituitary gland. This is the cerebellum, and this is the medulla oblongata going into the spinal cord. Why I'm showing this to you is just to show you a comparison. Perhaps that's what I stopped last time, telling you how the mouse brain, evolutionarily expanded into the human brain. And in this sagittal view, I'm going to show the same thing, that how a mouse brain looks very different from a human brain. Though the essential features are very same. So what I would like to really focus is, forget the cerebral cortex here, but if you look at the other features like the cerebellum and the medulla, and if you look at the hypothalamus, there is one thing which, if you can immediately look at, you will say that the cerebral cortex has definitely increased in size. And because it is increased in size, what it has done here is, from the mouse to the human, is that it has grown so much that it has covered the entire other regions of the brain. And because it had all had to be compacted and placed inside the skull of the human brain, it took on this structure. So basically what happened, if you can see me, is that this cerebral cortex, which was like this, grew all over the place, and then it got fit into the entire skull. So this cerebral cortex, assume that it grew on all sides, the lateral side, the posterior side, all over. If it grew, and then it will just grow on to cover the entire brain. And that's what happened in the human cortex, right? So just a bit of revision. What really is happening during development is that it starts with this tube, then forms a three-vesicle stage, where the very tip forms the forebrain, followed by the midbrain, hindbrain, and the spinal cord. So today, this three-vesicle stage soon becomes a five-vesicle stage, where these are the ventricles, or these are the hollows which are filled with the cerebrospinal fluid, which keep baiting the developing neural tube with signaling factors and everything. And that's what drives the entire development of the nervous system. So these ventricles at the very rostral end, they give rise to the telencephalon, which gives rise to the cerebral cortex, and so on and so forth. So for my today's from now, when we talk about divisions and all, what I'm going to really focus on is the cerebral cortex, or this very rostral end, where, you know, this is the ventricle, and the zone very close to the ventricle, it's called the ventricular zone, or that's the zone which consists of all the progenitors, right? Okay. So now that we've briefly discussed what all we learned in the last class, I mean, I'll probably just take a pause here before I go on to my, is there any way I can exit? Okay, are there any questions at this point? If not, I'll probably just proceed. Okay. So up until now, I told you how this entire structure comes about in development. What starts with the tube forms different, it's a hollow cavity, and then that forms different structures, embryonic structures, which grow on to make the nervous system. Now, one feature of this entire brain or the nervous system is the fact that it's an emerging function, is that things start very molecularly, then they go on to form cells like the neurons or the glia, which you see in the entire brain. Then that leads to the formation of connections or circuits. That leads to the, that is the underlying principle for the physiology. For instance, if it's a neuron, it needs to have the neurotransmitters to fire. If it's the glia, they need to have all the support factors which need to support the neurons. And that combination of cells going to circuits to the underlying physiology leads to the formation of the entire brain, which leads to the perception, or which leads to things like perceptions, like, whenever you see something, the image forms on the retina, which then carries all the electrical signals back to the brain, where a sense of it is made. Those neurons from the cortex then connect to the region called the thalamus by circuits. There are neurons in the brain, there are neurons in the thalamus, which connect with each other. They have an underlying physiology, and that's how you have things like perception. And once you have perception, you act on it, and that becomes behavior. So the emerging function in the nervous system starts from cells to connections to leading to behavior. Now, when you look at how the neural network is built, you will appreciate the fact that it's whatever you want to study as the structure in the adult, as a function in the adult. It's very important to study the structure or how it is built during embryonic development, because that gives an idea of how the system is built and how it may function. And the parallel which I showed you, the emerging function, you can see that a similar parallel exists during development as well. So the stem cells or the progenitors, or as I told you, all the stem cells which line, so there are these hollow ventricles, and whatever lines around those ventricles are the stem cells. Those divide. And at some point, at the onset of neurogenesis, they undergo asymmetric division. I'll come to that in a bit. The stem cell, if it divides symmetrically, it gives rise to two stem cells, and that expands the pool of stem cells. But that's not the whole idea of nervous system. You need to start making differentiated progeny. So those stem cells will undergo asymmetric division, whereby they'll give rise to one more stem cell and a neuron. And as in development progresses, initially it will give rise to neuron, and later it will give rise to glia. So that is fate specification. That is the second step in the building of the neural network. Once these neurons in glia are fate specified, they need to migrate. So this is, they are produced close to the ventricle, but they keep migrating towards their regions in the developing cerebral cortex. So neural migration is also very important. Once they migrate and sit in their place, they need to give out the axon or the outgrowth, which leads to formation of connections or circuits. And finally, that leads to the formation of a functional network, which will help you in perception and behavior. So the emerging function is actually a property of the way the neural network is built. Right. So as I was telling you, I'm going to be focused on the very, very, forebrain, the region where, all your, basically focused on the neocortex, and now tell you what are some of the properties of these stem cells, how they divide. And from there, take you into looking at how some of these conserved mechanisms have been studied in the mouse neocortical development. and how that is now with additional model systems like the human fetal brain and the human organoids, we've now gone into understanding how perhaps the neocortex evolved from the mouse to the human. So from now, my talk is going to focus on the neocortex, the divisions in the neocortex, and how those divisions change from the mouse to the human, right? This is a cross-sectional, this is a coronal view of the developing mouse brain. So it's cut like this in a developing brain. And this is shown across a developmental age in a mouse. This is here at the very end is embryonic day 10.5, and perhaps what I'm showing you is 2-3 days in development. So this is 10.5, this is around 12.5 and 13.5 here. But this slide here is to show you what are the different kind of cells, progenitors, which exist in the mouse neocortex. So at the start of 10.5, where neurogenesis, or the formation of the 6 layers of the neocortex, starts perhaps at the border between 10.5 and 11.5 in the mouse brain. That is, when I say embryonic day 10.5, 11.5, what I mean is the fertilization happened at day 0, and how we take it is we always calculate the midnight as the day at which fertilization happened. And when we always look at the vaginal plug, when we see whether the mating has happened or not, that's the next day. So we count a 0.5, assuming that it happened 12 hours before. That's why I call it E10.5. At several places, you will see that it's also called embryonic day 10, 11, and so on. So this is around embryonic day 10.5 here, where the primordium, so it is still not called the cortex. It's still called the cortical primordium, neocortical primordium, because it's still developing. So the neocortical primordium here consists of neuroepithelial cells. And what I would like to show you here is, unlike the stem cells in the Drosophila, these have a very unique structure in that they have apical process. Sorry, is my cursor? Yes. So this is the ventricle here. Here, this is the one, the region below this consists of the ventricle, or the cavity, hollow cavity. Just right above the ventricle is the ventricular zone, where all your progenitors, or something which is very similar to the neuroblast in the Drosophila, they are present in this ventricular zone, right? Then this is followed by another zone of progenitors. It's called the subventricular zone. And these are more restrictive in potential, in the sense that the potential to divide and generate all cell types of the brain exist only in these apical progenitors. And that becomes restrictive as the cells go from the ventricular zone to the subventricular zone. And this is the intermediate zone where the neurons are migrating. I'll come to that in a bit, how they migrate. And this is the cortical plate. The cortical plate essentially consists of differentiated neurons, which now are forming the growing cerebral neocortex. So what I really want you to appreciate here is this. This is the apical end, and this is the basal end in the developing cortical primordium. And these neuroepithelial cells and the radial glia, which are derived from them, all have an apical process and a basal process. And the basal process is sticking out to the PL surface. This is a surface on the top, whereas the apical, it's all connected from the top to the bottom. And I'll come to in a bit that how it is useful. So at 10.5, this entire primordium only consists of neuroepithelial cells. These undergo a lot of symmetric divisions, so that before the onset of neurogenesis, you have enough stem cells which can generate or last your entire cortical development. Just to give you an example of how, if this system is compromised, you end up having microcephaly, where it is a disorder in which you actually have a small cortex. It's purely because there is a mutation here in the division of these neuroepithelial cells. And because of the mutation, what you end up having is the proliferation is affected. You have a small number of pool of stem cells, which again give rise to all the different kind of neurons. That's not affected. But what is affected is the number of stem cells which were to begin with, which could have generated the entire cortex. So this is a very crucial step, the symmetric division of this. So these neuroepithelial cells then become something called an apical radial glial cells. Now, these are those cells which now have the capacity to undergo both symmetric and asymmetric division. These undergo symmetric division so that again their numbers are increased so that they can also sustain development. But they also undergo an asymmetric division where they give rise to two daughter cells, one of which forms a neuron, the other forms another radial glia. So that is an example of a asymmetric division. And then these radial glia, so this is whenever it gives rise to apical radial glia, which is a progenitor and a neuron, that is called direct neurogenesis. When directly these progenitors give rise to neurons, that's called direct neurogenesis. These apical radial glia can also give rise to another progenitor subtype called the basal intermediate progenitor. The reason they are called basal is simply because they don't have that apical process and they migrate towards the basal end of the cortex. So these are called basal intermediate progenitors. Now, as I said, these progenitors are restrictive in their potential in that they only undergo one or two divisions maximum. And sometimes, in fact, they directly divide and form two neurons. Or in fact, they may undergo one more division and form neurons. Now, all of these neurons which are formed in the cortex, they use the the basal process of the radial glia to migrate. They say they migrate along. It's as if the radial glia guides them through the cortex and tells them that you need to migrate here and go and sit in the developing cortical plate. These apical radial glia also undergo a different kind of division, whereby what happens is when they divide, they leave off their apical process, but still continue to have their basal process. And that is called a basal radial glia. Now, if you look at the difference between a basal radial glia and a basal intermediate progenitor, it is that these still hook on to their basal process, whereas these don't have either the apical or the basal process. These again can quickly divide into basal progenitors or make lots of neurons. And then all these neurons migrate past these basal processes and sit here, right? So the steps in the development are like neural epithelial cells forming apical radial glia, which then forms your basal progenitors, which are either the basal intermediate progenitors or the basal radial glia. Then your different neuron subtypes are formed, which then mature or they migrate and their dendritic morphogenesis happen. And I'm going to explain some of these steps in detail so that you can understand how it happens during development. So as I was telling you, and now I'm going to tell you a parallel to which I think perhaps Sonia has also shown this to you, that how some of these are very similar in the Drosophila. So the apical radial glia undergo symmetric division, that's the proliferative phase, and this is in a gestating human brain when this happens. Now, a form of direct neurogenesis is that when it directly forms a neuron and apical radial glia, that is direct neurogenesis. But, and I think perhaps this is called type 0 in the Drosophila, and this here mode is called the type 1. And you will really appreciate in me telling that though this mechanism is important, but it is not enough to sustain the entire cortex. So if the cortex were to only undergo direct neurogenesis, then probably you'll not be able to generate enough numbers. So basically, all these kind of divisions have perhaps come in evolution simply to accommodate for the expansion. I mean, or because of these divisions, you have expanded your neuronal pool, going from simple organisms to more complex organisms. So this is the type 0 as seen in the Drosophila, and the indirect neurogenesis, which I told you, is when apical radial glia forms the basal intermediate progenitor, which can undergo one or two division or directly form neurons, or the ARGs becomes ORGs, which can again give rise to neurons and ORGs, and that then sustains the neurogenesis. And I'll come to in a bit that how the expansion of this ORG has given the neocortical expansion. I mean, a quick check before I move on. Are there any questions for me till now?
What is ORG? Sorry.
Yeah. So they are, you want the expansion, or it's called outer radial glia, because
Okay.
just 1 minute. Yeah. So I'll come to that when I talk more about the human development, like why are they called ORGs. But I'll just, maybe since you asked, I'll show it here. So if you see that this is a ventricular zone here, right? Now this is the subventricular zone here, right? In case of humans, there is an additional zone which is above the subventricular zone, which is called the outer subventricular zone. And the ORGs are very specifically, this is a mouse brain, but so the OSVZ is not very quite visible, and there are very, very few BRGs in the mouse brain. There are hardly any. I mean, it's thought of that the basal radial glia or the, so the basal radial glia are also called the outer radial glia. So in the mouse, there are very, very few. The thought process in the field is that perhaps it was in evolution, it was there in the mouse, but a few, and it just expanded in the humans, and it gave rise to an entire new zone called the outer SVZ, which I'll show to you when I talk about the human brain development. And that, because they are the outer radial glia are in the OSVZ, they are also called the outer radial glia. Is that okay?
Yes, thank you.
Okay, yeah, right. So, okay, what I'm showing you here is a division from the, I'm going to show you a video also, but this is just a screenshot here. So this is the ventricle here, and this is this apical radial glia, and this is the developing primordium, right? So this is, as you can see here in the apical radial glia, I'm just trying to show you some of these divisions, and I'll show you a video also where you'll immensely appreciate how these divisions happen between apical radial glia in a basal progenitor and how the daughter neurons are generated. And I'll ask you to hold on the ORGs. I'll talk about the ORGs during human brain development. Okay. So this is basically the apical radial glia. As you can see that, the process is here. The process goes up till the pile surface as well. And this is the cell body of the apical radial glia. And these are the neurons which have been generated from this apical radial glia. And in this case, perhaps by direct neurogenesis, and they migrate across this. And here you can see that, this is a video, and I'll show you a video as well, that, so one of the characteristics of the apical radial glia is that it undergoes nuclear migration. It's called INM, where this nucleus will keep migrating from the apical to the basal surface. And this happens during the cell cycle. As it will divide, always the nucleus will always be here in the apical surface when it divides, but as the cell cycle progresses from G1G to G1G to phase, it keeps migrating, and it comes back during division. So shown here is the dividing apical radial glia, and you can see here that it's dividing. It gives rise to neurons which migrate past, and here you can see that the neuron has absolutely taken the shape of a bipolar structure. You can see, so these are called pyramidal neurons because their cell body has a triangle shape, a pyramid-like shape. And you can see here again that these are migrating past this. So I'm going to stop this share here and show you a video. And this video is from, do let me know if you can't hear the video. And this video is from Arnold Kriegstein. And here I must say that some of these beautiful videos of how these apical radial glia, the basal intermediate progenitor, in fact, his lab is been instrumental in identifying the plane of division, or how these divide, and the existence of basal progenitors and outer radial glia, both in mouse and humans. So he has a video on the iBiology.org website. I highly recommend you to watch it, but I'm going to show you a portion of the video so that you can appreciate how these cells divide. Can you hear the audio as well? Hello?
No, maybe it'll help
No,
if you
we can't.
increase the volume.
Okay. If not, I can do a voiceover.
And it confirms what we sort of
We can hear.
hypothesized from those still images. The first cell that was initially infected was a radioglial cell. And as shown here, it divides at the surface of the ventricle and produces a daughter.
So did you just see that it undergoes a vertical division, right? These apical radial glia? Yeah. Yeah.
But the
And-
radioglial fiber never retracted. So that radioglial cell was a self-renewed radioglia after that first division. And then the nucleus of the radioglial cell moved back up. It goes through interkinetic nuclear migration, as do all neuroepithelial cells. And the daughter cell started migrating away. And I hope you can appreciate it's migrating along the parental radial fiber. And initially,
So what I really want you to appreciate is that, as you can see here, the apical radial glia had underwent a symmetric division. I'll come to that in a bit. And the daughter cell migrated, but also the nucleus of the apical radial glia itself was moving. It was that's called kinetic nuclear movement. That itself was moving. But if you had observed that as the daughter cells moved away from the use of fibers to move away from the parental radial glia, the nucleus came back to the surface and came back to the surface to undergo another round of division. And that's how it happens in the developing neocortex.
thought these could be
Bhavna,
neurons.
Yeah.
But
So yes.
so what is that? What is that process? Is it cytoskeleton?
Yes, yes, yes. So yes, it is microtubules and yes, that's the cytoskeleton. You're right.
Okay, and nuclei just sort of climb up and down. What?
Yes, yes, they do. That's yes, the nucleus just goes up and down. I thought I may have some schematic to show that, but maybe not. But yeah, so this is, in fact, it is thought that the internuclear, so this migration has, I think it's still not understood, what is the function, but there are two things associated with this migration, this internuclear migration. I mean, I think you're surprised at this, but wait till I show you the ORGs and how they jump during their division. So that is really fantastic. And I'll come to that towards the very end. So the two things about this internuclear migration is one, it happens across cell cycle, which means every time it has to divide, the nucleus comes back to the apical surface. Now, another important thing is, it's also thought to be important for the nucleus to sort of guide the daughter cell across the parental fiber. So I think it is associated with that, the migration plus the cell cycle. So these are the two things which I think this migration does.
Okay, and
Sort of
is it known what the reason for it is, that it travels a certain zone only? Is there something that it's picking up from there, or?
So it is traveling till the ventricular zone. So maybe, okay. Can I come back to that? Let's just finish this video and I can come back to that.
Yeah,
Can you
sure,
just
sure,
hold
sure.
on to that thought? Yeah?
Sure.
Perhaps when I come back to my slides, I think we can have a discussion on to where it migrates. Yeah.
Okay, thank you.
Yeah.
Observe more carefully and notice that when the radioglial cell goes through its next cell cycle, which is shown here at the vent.
So I mean, you would have all observed that it did undergo one division. It went, and as I mentioned, the nucleus migrated. The daughter cell migrated on top of it. The daughter cell went on its path, and the nucleus came back, and it's undergoing another round of division here, right?
The daughter cell also rounded up and divided. So this wasn't a neuron. It was
Now all this was apical radial glia. Now I want you to focus on the this daughter cell, and he's going to tell you what this daughter cell now becomes, right? That becomes the intermediate or the basal progenitor.
actually a different type of progenitor. And so we call those intermediate progenitor cells. And that progenitor cell, shown here, divides symmetrically to produce two daughter cells that continue migrating up to the cortex and in the end become a pair of cortical neurons.
And also observe that at the same time as these basal progenitors divided and formed two progeny, which will go on to differentiate from neurons, the apical radial glia underwent another round of division at the apical surface. So this is how the division in the neocortex proceeds.
So this pattern of neurogenesis, where the radioglial cells turn out to be the neural stem cells, was really quite unexpected at the time, but has now been widely recognized as the way most cortical cells, in fact, most CNS neurons, are born from radioglial cells. Now those.
Okay, so how do I stop the share? Okay, where are my floating panels? Okay, sorry, I had to, just give me 1 minute, I'll be back.
UCSF to get donated tissue that we.
Okay, yeah, so we are back. Yes, Shubha, you had a question, right?
What is ORG?
Yes, so yeah, so the ORGs are the outer radial glia. Hello?
Yes, yes.
Yes. No, so right. So so the ORGs are the outer radial glia or the basal radial glia, right? And the division I didn't show in this particular video. I'll because they're quite expanded in the humans. I'll show you the mode of divisions in the human development and show how it is different from the division in the apical radial glia. So, but I think there was some, maybe you or somebody who would ask the question that, why is that this migration only happens in this zone? If you would see that this is the ventricular zone, or the zone where your apical radial glia exist, and the zone beyond this is now the intermediate zone. So the migration of this is only till in this particular area, where it perhaps, as I said, it's connected to the fact that it's basically guiding the daughter cell up into the developing cortical plate, developing cortical primordium. So that is sort of the reason why this migration is seen. I mean, I cannot say it with full certainty. Perhaps even I don't know that why this migration only happens in this particular manner.
So then, does something happen if
this is
if the
INP, yeah.
Yes, hi. So does something happen if the migration is not allowed to happen? Like I don't even know if such experiments would be possible.
Yes, no, definitely. So that is there. When you block the migration, perhaps this migration is aided by the cytoskeletons. If you block it, you'll end up having a whole bunch. So I think probably what you end up having is a whole bunch of progenitors which are stuck here and not able to migrate past this.
Okay.
Right. So yeah, so many things are affected, because this is one of the first step very important in the cortical development. All your neurogenesis and all is affected, definitely. But importantly, what happens is everything is stuck close to the ventricular zone. Nothing migrates. Nothing moves past this zone. And maybe I could have, you can just maybe read up on some papers on that, but you see that, it's all stuck close to the ventricular zone. The migration is highly defective.
Okay.
Yeah.
Thank you.
Bhavna, Sonia here. Is it legal for me to ask something?
Yes, definitely, Sonia.
So I guess one thing would be, if is it trying to escape some sort of a niche that forces it to divide symmetrically?
So niches are very well studied in Drosophila. What is it understood? I'll come to that. I think what guides the division and all, right? I'll come to that.
Okay, so if you block that division, they undergo more symmetric division. Is that what?
Yes.
Okay.
Yeah.
I think was it Jürgen's lab that showed that if you fiddle around with the same inscrutable business, you get more of these symmetric divisions.
Yes, maybe. So I'm not aware of the exact mutant which may cause this. I'm not sure about it. But I'll just come to a very wild type scenario where, you know, what are some of these factors which decide how the division happens. And maybe if you block one of them, I know that if you block par3, which I'll be coming to, if you can hold to that, that maybe we can have that discussion in that slide.
Yeah.
Of, yeah. Okay. So, right. So I showed you some of these divisions. Okay, I think I will come to this after I talk about, so, okay, my slide, it's just at the wrong place. Okay.
Bhavna, I have a question regarding
Yes.
the interkinetic nuclear migration and
Yes.
the significance of it.
Mhm.
So is it, so what is the simple function of this INM? Is it to maximize the number of mitosis at the apical surface so that there are more neural progenitor cells, or what is it?
Correct. So I think it is just, there are few things which people think of why this is happening. And one of the main reasons for this interkinetic movement is the fact that it has to now, I mean, see, the daughter cells have to migrate past the fibers, right? So I think one of the functions of this internuclear interkinetic migration is that it guides the daughter cell across the fiber. And as you would see in the video also, if you remember that, you know, the nucleus was migrating and on top of it was the daughter cell. It was as if this was nudging the daughter cell to, you know, migrate past the fiber. And that there is an observation that this is then also connected to the cell cycle, because the division always happens at the apical end, but not at the any other end. So the division happens and then all the other cell cycle phases happen, and then it comes back to the apical phase to again divide again. And your thought process on whether what was that you mentioned? I think that's also one of the reasons. Yojit, could you just say that?
Yes, to maximize the number of
Right.
the
I mean,
progenitor.
maximize the number of progenitors, or the fact that every division has to happen apically. So if a cell is not undergoing division, so perhaps it's just moving out so that, you know, another cell can divide at that place. I think it's just a hand waving thing. I'm not sure if that's one of the reasons. I think one of the main reasons is that it has to guide the daughter cell across the fiber. Yeah.
Could it be like the reason why these go up and down is because like as it goes up, there are different gradients that are formed, right? So once it divides there and leaves a daughter cell there, that daughter cell is destined to become one subtype of neuron compared to other. Could that also be the case?
Could that be the case for the migration?
Yes. Like the function of INM.
Well, I don't know. I don't. So I don't think the migration is connected to that. I will perhaps tell you a bit about how the different neural subtypes are generated. I'll come to that, because some single cell RNA-seq studies show that what. So I think if I may phrase your question as two separate things. One, I don't think the migration is connected to the generation of the different, or there is not yet known, or people have not looked at it from that viewpoint, whether the migration is connected to the different neural subtype identity produced.
Okay.
But the temporal specification has a cell intrinsic role, and I'll come to that.
Okay.
Yeah, that has a strong cell intrinsic role, and
Yeah.
I'll come to that. Yeah.
Sure.
Yeah. So, I mean, I think if I may just pause here and mention this, I don't have slides for that, but now that you brought out that point, is okay. I'll probably take it up when I talk about the cell intrinsic role. I'll take it up there. Okay. Now coming to the division itself. So the division, as you would all seen, that if a symmetric division undergoes apical division undergoes a vertical plane of division, whereas an asymmetric division is horizontal. And this was thought, Sue McConnell did these studies, and they saw the divisions were bang on, like perpendicular. But Weiland-Huttner's lab showed that these some of these divisions are not perpendicular, but actually in an oblique manner. The division actually happens in an oblique manner. And it's thought, which I also don't understand, it's perhaps to give the apical membrane some space or something. I also don't understand. So if any of you are interested in knowing why it is oblique, I recommend you to study some of these papers from Weiland-Huttner's lab as to how this happens. Now, coming to what could, so it is thought that this division and all of these are controlled by several factors. There are potential intrinsic factors. For instance, I think Sonia discussed the PAR3 and the NUM proteins, whose localization decides the plane of division and the kind of division. And also, if you have defects in PAR3 or NUM, you end up having more symmetric divisions as opposed to asymmetric divisions. And some of these migration defects, if you if you knock out some of these cytoskeletal proteins, you end up actually the migration getting affected. You make the basal progenitors, they don't migrate. They all populate close to the ventricular zone. So those are some of the defects which you see when you tinker with this process. Now, besides the cell intrinsic factors, there are two other main factors which regulate this process. One is extrinsic factors, and these come from several sources. And I like to mention two important sources. One is, of course, the cerebrospinal fluid. In all of this, I think we should highly appreciate that there is a fluid underneath all this growing epithelium, which keeps baiting the epithelium with some of these factors like Wnt, BMPs, FGFs, and IGF2. And if you tinker with any one of them, some of these very early divisions are indeed affected. And so you also have these blood vessels, which bring some of these extrinsic factors. You also have interneurons. Now, one of the, so there are two main neuronal types in the developing cortex. First is the excitatory neurons, the other is the interneurons. The excitatory or the pyramidal neurons all come from the process which I just defined to you, come from the apical radial glia in the cortical primordium. But the interneurons have another source. They all come from the ventral. So from the forebrain, the ventral side of the forebrain. So these neurons are also developing at the same time as your cortical pyramidal neurons are. And then they come up from outside of the cortex into the cortex, and they bring sonic hedgehog with them. So that is also something which is happening simultaneously, and that could also sort of affect, is known to affect these divisions. Besides, of course, I'll be coming to this reelin molecule in a bit, that there is on top of all this growing epithelium is a zone called the marginal zone, which has another cell type which secretes something called reelin, which is a signaling molecule, which is very important for these migrating neurons, for them to migrate. In the absence of migration, and I'll be showing how the cortex itself is inverted in the absence of reelin. So these are some of the factors which decide the division. It's both cell intrinsic and both cell extrinsic. Okay. So what I defined to you was the first few steps of making the different progenitors. The apical, the neural, it starts from neural epithelial. So this is showing you the entire development from embryonic day 10.5 to postnatal day 4. When you can see here that this is the 6-layered cortex here. Oh, sorry, it's 10:30 and I'm still probably not even midway. Okay, let me just quickly go through. So this is the 6-layered neocortex here, and you can see here that the layer 6 is here, layer 5, layer 4, 2, 3, and 1. And how this comes up in development is, to begin with, at 10.5, it's only neuroepithelial cells. They can undergo direct neurogenesis, make some neurons, and this is called, they do make some initial neurons called the preplate neurons. Then they become the radial glia, apical radial glia, which also undergo neurogenesis direct, or give rise to a basal progenitor, which gives rise to a neuron. So at embryonic day 12.5, what happens is whatever neurons they come, they form the first layer of neurons, that is the layer 6. By 13.5, the same apical radial glia, they make basal progenitors, which now give rise to neuron, which go on to form the layer 5. So they migrate past the subplate, this layer 6, and go and sit on top of this layer 5. So there is, of course, this division. There is this migration of the daughter cell through the radial glia, and then there is migration through all of these layers and going and sitting on top, and so on and so forth. So you make layer 6, then you make layer 5, which migrates past layers 6 and sits on top. You make layer 4 then from the same progenitors at 14.5. The layer 4 will be formed, which will migrate past, layer 6, 5, and sit on top. By the time you also have some outer radial glia. So it's the expanding cortex is because the expanding cortex is then produced both from the outer, the basal or the outer radial glia and the intermediate progenitors. So then you make, yeah.
Hi, Bhavna. I had a question.
Yes.
Is it known where does the cell spend energy in moving past these already existing layers? And I would have thought it is more economical to just form like the closest layer, stay closest to where it's being born, rather than move through all of these layers and go past it.
So that's a wonderful thought. I actually, to be very frank, I don't know the answer for that. So the fact is that this happens. So it's an so let's look at it this way. It's an observation that these cells migrate past, right?
Mhm.
So which means that it's an energy expensive process. So indeed, the neurons and the glia are actually, I mean, the brain takes up most of the glucose from the body, right?
Yeah.
Yeah. So indeed, it's an energy expensive process. So perhaps now we need to go back and look at what, I mean, I think it's sort of a chicken egg question, that what came first, whether the migration happened and then the cells adapted to the increased use of energy, or they were already using a lot of energy. And so I think it's a combination of mechanisms, and I'll come to in a bit that why this happens. I think renin is sort of important molecule which determines the migration past. And maybe an answer to that will be the fact that, so let's look at it this way. For this to migrate past layer 6 and 5, you probably need to have an attractant Q, say, sitting here on top where my cursor is,
Mm-hmm.
right?
Yeah.
Suppose if the attractant was here, say at the subplate, the cells would just migrate and sit here.
Yeah.
Right? Now, actually, it turns out the attractant is sitting here, and I'll come to that. What is that attractant?
Okay.
Since the attractant is here, the cells are all migrating past the previous layers and sitting on top of it.
Okay.
That's perhaps one way to understand that why this migration would have happened, or why this, going past would have happened at all.
Yeah.
Okay. So, yeah, Khushbu, do you have a question?
No, sorry, it was a mistake.
Okay, yeah, no problem.
I kind of had a follow-up question as
Yes.
to why does this have to happen? I mean, are these cells different? Why does
Yes.
this arrangement matter?
Yes, I'll come to that.
Okay, sorry.
Yeah. Why is this arrangement important? Yes. I mean, to give you a very quick answer is that all these neurons are very different. So this, anything which is in, say, green is called a corticothalamic projection neuron. Anything which is in, say, red is a subcerebral projection neuron. And I'll come to that, how each of these neurons are different.
Okay, thank
Yeah.
you.
So it migrates and it goes and sits here, and bang on, your answer is here. So what are these different neurons? The layer 6 neurons, or the corticothalamic projection neurons. Now, as I said, the brain is highly organized. Organized. That anything which comes from the outer world doesn't go directly to the cortex. It's not as if you saw something and then you directly it got fired in the cortex. It is highly organized because any input which comes from the outer world, be it your vision or your hearing or your touch or your taste or anything, it comes to a relay center in the brain, which is called the thalamus, and which is here, which is marked, this nucleus here is marked as the thalamus. Now this thalamus gives outputs to the brain, right? And I'm not, this output is not that, but it gives output to the brain, which goes to the layer 5. That's called the thalamocortical projection neurons, which is not shown here. But once the brain decides on some action and it wants to tell your hand to move, when it, say, if it sees a ball, you want to move your hand so that you can, chuck the ball or something, or, or catch the ball if you were a cricketer. What neurons it uses to do that are the layer 6 neurons, which are the corticothalamic projection neurons. So it comes from the cortex, it projects to the thalamus. So as you can see here, it is all these green ones are projecting to the thalamus, and from the thalamus, the neurons go to the spinal cord, and then they go to the different parts of the body. And that's how the decision from the cortex is relayed to your body, right? So those neurons are exclusive of layer 6. Now, when you look at layer 5 neurons, which are just above the layer, so layer 6, these are your subcerebral projection neurons, which directly project to your spinal cord, and they are important for your movement. Your walking and all this, anything to do with muscle movement is all directed by these subcerebral projection neurons. Now, you are a brain, your left hemisphere and the right hemisphere have to talk to each other, right? If you have a cut in between the two hemispheres, you cannot have all these higher order functions, because your two hemispheres constantly talk to each other. You have some cognitive deficits where your one hemisphere is not talking to the other. You may have some, you may talk well, but your language is processed on one end, but your thought process is on the other end, right? So you may have a disconnect, but you may be able to speak, but you may not have, concerted thoughts. So that's because of these callosal projection neurons, which come from the upper layers, the layers 4, 2, 3. So these project, as you can see here in blue, these are coming from your upper layers, layer 2, 3, 4, 5, and they are going to the other side of the hemisphere. So all these 6 layers have different architectures. They are different, and try to understand from that emerging function, which I told you from the very first slide. The neurons give out connections, and that decides the function. So this is coming in that, that the neurons, how they come up in development, they then give out the projections, and that then decides the function of this particular neuron in adulthood. Is that clear, Meghna?
Yes, thank you.
Okay.
But so all the formation of the cells, I mean, it's happening in phases, right? So
Yes,
it is kind of clockwork, like it is
correct.
decided at this time these kind of cells will be generated, at the next phase these kind of cells will be generated.
Correct.
So
I'll come to that clockwork, the temporal, very nice word actually. We use temporal specification, you've used clockwork.
okay,
Yeah, so I'll come to that. I think if you can hold, I'm already, I'm just going to take
Sorry.
some, no, very good. I mean, I'm happy that, these are triggering a lot of questions, and some of these questions have triggered in me as well. And I think my lab is focusing on answering some of these questions. So good. So hold on to those thoughts. I think I'll be able to answer some of those as we go along. Yeah. Okay.
Sorry.
So
Yes.
let's just quickly move on. I think I have a lot of slides to cover, but a lot of interesting things. Is it okay if I overshoot time by a little? I'm not sure what your lives are after 11.
We have class at 11 today.
Ah, okay. So, okay, let me just try to, let's see where we are, and maybe I'll just give you my slides. So, Varsha, how do we do this then? Should I just finish my slides, and maybe if people have doubts, they can contact me? Hello, sorry. Did I lose Varsha?
Bhavna, if you feel, we could ask Raj whether we can extend by one lecture. Would you like that?
Yes, actually, I would like that, because I I do have a lot of slides, and I'm most of the interesting stuff is still coming.
I'll
Actually.
talk to Raj after this.
Okay, so maybe I'll take a pause at some point.
You go as you would.
Okay, great. And I'll stop at 11 and then maybe I'll take another class after this.
Yeah. Okay.
Yeah, okay. Okay. Oh, sorry, it's doing some auto recovery. Okay, that's done. So, okay. So these are the different neurons in the adult cortex. And as you can see here, this is layer 6 marked by blue. This is layer 5A. This is layer 5B here marked by CTIP2. This is layer 5B. This is ROR beta layer 4. And these are all the layers 2, 3, which are the upper layers. And as you can see that the upper layers are actually more than the deeper layers. And perhaps this can then connect to the fact that your basal progenitors and ORGs come up later in development because the expansion is more in the upper layers. So you need more different kinds of progenitors producing a lot of neurons to sort of, make up for this. And this is important because whatever expansion, whatever upper layers you're seeing here is hugely expanded in the humans, right? So, okay, here shown is another image as the development progresses that, what are the different layers here marked with 6. So this is staining of the markers plus BRDU. So if you would pulse BRDU at different ages, what you would actually end up getting is marking several of these different layers, layer 6, 5, 5, 6, 5 here, 4 and 2 and the, okay. Now this zone here is called the marginal zone or the zone where your another cell type exists. Let's not go to that. That's another cell type which comes in development called the Carl-Radius cells. What is important to know is that these cells produce a signaling molecule called relin. And I was telling you in my discussion with maybe IMT that, the signaling which pulls the neurons or helps or tells the neurons that you need to migrate is this signal. Now, if you don't have this signal, what happens is this. So this is a reeler mutant. This is a naturally occurring mutation in relin which people saw. And when they looked at the brains of such a reeler mutant, they saw an inverted cortex. Now, here is a control cortex where TBR1, which marks layer 6, is here, and this is layer 2, 3. But in the reeler mutant, you would see that is just completely opposite. The 2, 3 are here and the layer 6, which means the migration past those layers has not happened. So the cells divided, made daughter neurons, it couldn't migrate because the signaling center, which was on top, which was telling the neurons that you need to migrate past, and that was a chemoattractant was missing. And not just the cortex is inverted, but the projections are also changed. And that's shown here is, if you would see, this is the control cortex. And this is again those thalamocortical neurons, which is coming from the thalamus, which come and sit in layer 4 here. Sorry, these are the corticothalamic projections which come and sit here. But as you would see that now, since the layer has gone on top, the projections are now very close to the pale surface, right? So not just, identity. So this is the emerging function, and this is why neural development is very, very important, because whatever gets specified also gets connected. So if you specify it wrongly, or if you misspecify it, you end up having wrong connections, and that's what leads to, perhaps is the basis for several of these neurodevelopmental disorders, right? So coming to the question of what times or sort of what is the temporal timing of fate specification. Now, I must here mention that one of the easiest ways to understand this, whether it is cell autonomous or extrinsic or coming from outside, is simply to take some of these stem cells and put it in a dish. If it is cell intrinsic, you would still have the same clockwork, and I really like that word, so which Meghna used. So you'd have the same clockwork as you would have it in the developing brain, and this is what happened. Sally Temple, in some of her classic studies, she took out some of these progenitors right from embryonic day 10.5, 11.5 and put it in a dish. And she asked the question that now I'm not going to give it any extrinsic cues. Is it intrinsically driven? Or do these progenitors know they have some clockwork inside telling them that, These are the number of divisions I've undergone. I need to first produce layer 6, 5, 4, 3, 2, and 4 and 3, 2, 3. And that's what happens. So if you take some of these progenitors, put it in a dish, you get layer 6 first, 5, 4, 2, 3. And then once all the neurons are made, you actually start making the astrocytes or the glia. And even that happens. So which means that it is largely cell intrinsic driven, and extrinsic cues only shape up some of these, maturation effects, like how do the neurons mature and all of that. So coming to the question of temporal timing, then it is a combinatorial expression of transcription factor programs which define what kind of projection neurons will be formed in these progenitors. And there are two ways in this, in fact, four ways in which this gets decided. The first decision always happens in the ventricular zone progenitors. That is the progenitors which underlie the ventricle. So the ventricle zone progenitors, some have already been characterized, and I'll be talking about my own work on LHX2. That expression of factors like EMX2, PAX6, SOX6, SCOOP-TF1, NGN2, and FESF2 and LHX2 determine the fate of these progenitors, like how these progenitors, the expression of these transcription factors or a combination of these determine what is the sort of neuron it will produce during neurogenesis. Once these progenitors become slightly fate restricted, become SVZ, then their expression also determines the kind of neurons which will be formed, which is expression of TBR2 and CUX2. For instance, if there is TBR2, they give rise to all the layers of the cortex. But CUX2 lineage is known, the progenitors, SVZ progenitors, which are CUX2 positive, they only give rise to the upper layers. So here itself, you can see that, that this distinction with the expression of certain transcription factors decide what will be the fate of the coming neurons, right? So for instance, like here itself, FESF2 is again restricted only to the deep layers. It's got nothing to do with the upper layers. And CUX2 expression is both in the ventricular progenitors and the SVZ decides the upper layer fate. Now coming to once they become just post-mitotic, then itself, once it happens that some of these transcription factors like PAX6, they're only expressed in the progenitors and they're completely switched off from the post-mitotic progeny. And in such post-mitotic progeny, you begin the expression of some transcription factors like FESF2. Now, FESF2 is very important for making of layer 6, and I'll come to that in the next slide. And probably I'll stop my talk today with that because there's just too much information overload will happen, and then maybe take up from there if I can get an additional class, and I'll ask Raj about it. Right. So as I was saying that once the progenitors become post-mitotic, they switch off some of these expression of EMX2, PAX6, EMX1, SOX2, and they become post-mitotic and they begin the expression of, if they are making a deep layer. So if this would be the embryonic day 11.5, 12.5, 11.5, it'll express TBR1 and SOX5. If it is 12.5, where you need to make layer 5 neurons, it'll start expressing FESF2, and I'll show you that. And when it is up, say layer 4 and 2, 3, it's BHLHB5, and its upper layers, it's CUX1, 2. Now, once they become just post-mitotic, there are these transcription factors, and once they become mature, and new set of transcription factors come up and seal the fate. So it's like that these markers just say that you need to be a deep layer neuron. But once they migrate and they sit, they migrate and sit in that layer, say they migrate past a previous layer and sit on top, there also many changes keep happening as they become mature, as they give out their axons. And another set of transcription factors come up and regulate this process and mature the neurons, give out the axons, make the synapse, and all that happens, right? So as I was telling you, I'll stop with this switch, which I have studied and which is sort of known in this. I'm going to be talking to you about one such switch where the regulation switches from layer 6 to layer 5 production, and I'll stop with that. So the layer 6 are the corticothalamic, which project to the thalamus, and two transcription factors, TBR1 and SOX5, are very important for making these neurons. And when they're making these neurons, they make sure that layer 5 does not get produced. So they inhibit the expression of transcription factor FESF2 and make sure that, it's only layer 6 production. But at the onset of layer 5 production, two other SOXs, SOX4 and SOX11, literally knock off SOX5 from the promoter and the enhancer region of FESF2 and activate FESF2. Now, what does this do? Activation of FESF2 in turn goes back and represses TBR1. So which means now the switch has been affected. So once you repress TBR1, TBR is now no more able to repress FESF2. FESF2 gets activated by SOX4, SOX11. It's both an activation by SOX4, SOX11 and the relieving of the repression by TBR1, which then switches the fate from layer 6 neurons to layer 5 neurons. And then this activates CTIP2, and then the layer becomes a layer 5 neuron. And I'll not talk to you about what happens with SAGB2. Just forget that bit. Okay. Now, I just wanted to put this out here because I have studied one such mechanism, and I showed to you how LHX2 is involved in this. Is that LHX2 actually regulates the expression of FESF2 and SOX11, and this happens inside the progenitors. It recruits these chromatin modifiers onto the enhancer and transcription start site of FESF2. And represses FESF2 and SOX11 expression. So when you don't have LHX2, you have active marks, the histone marks going up, the FESF2 and SOX11 is upregulated. And in the mutant of LHX2, what I saw was the expansion of layer 5. As you would see here, this is a control layer 5, and this is a mutant LHX2 layer 5. You see that the layer 5 is expanded at the expense of layer 6. And this is what happens if you knock off certain transcription factors, and these transcription factors regulate the timing and regulate, so what LHX2 is doing is it's regulating the timing of, so in this web, LHX2 is regulating the timing of layer 6 to layer 5 production. So at the time of layer 6 neurogenesis, you definitely need to keep FESF2 and SOX11 repressed. So I told you how if SOX11 was expressed, you would get activation of FESF2. LHX2 is keeping SOX11 repressed, which then in turn cannot activate FESF2 at the time of layer 6 production. And this is one of the switches which I studied in this developing Primordium. And in my own lab, what we are actually now studying is how these chromatin modifiers could further, regulate this switch, because it's all cell intrinsic. It all comes from chromatin regulation, because you need to change these active marks and all of this at these positions. So I'm in turn trying to study how these three interact with each other to make the entire the molecular switches between these different layers. And with this, I think I'll stop. Do you have any questions for me till here? Or was it too much to take in today? Hello?
While people are warming up, can I jump in, Bhavna?
Yes.
Can you take us to that Sally Temple slide?
Oh, that's not a slide. I didn't have that data.
No, but the schematic, the 25. Yeah.
Yes. Oh, okay. Yeah.
So if when people study this kind of thing, I'm looking now at the ventricular zone progenitor part of it. So are these is that over time? So is it moving from EMX to PAX to SOX to COOP to NGN to LHX over time?
No. So over time, there is a change, and I'll come to that with the single cell RNA-seq data. So I think till now, whatever I've shown you is all from bulk RNA-seq, RNA-seq or bulk genetic studies, knockout studies. So what is, so they have gradients, like PAX6 and LHX2 have an opposing gradient. So LHX2 is medially high, laterally low. Medial is where the hippocampus is, and laterally is the neocortex. PAX6 is laterally high and medially low. So all these are bulk studies where it's across expression gradients. EMX2 has a very similar gradient to that of LHX2 and an opposing gradient to PAX6. COOP-TF1 has a very region-specific expression. It's perhaps expressed only in the somatosensory areas. So whatever studies I'm showing you till now, it's bulk-seq. But I think I'll show you, and I've stopped that, and my next slide is going there to the single-cell RNA-seq to say that, how is this changing across time? So when it's a progenitor, I'll show you data to say that when it goes from embryonic day 12 to 15, the architecture or the expression changes completely of this progenitor itself. When it goes from E12, it becomes introverted to become slightly extroverted. So I'll come to that data. So all of these studies are from bulk-seq and from genetic studies, from gradients and all of that.
So these are likely to be more spatially organizing factors, regionalizing factors.
Sorry, Sonia, your voice got cut, so I couldn't
Can you hear me
the question.
now? Can you hear me now?
Yes, these are regionalizing factors. Yes, you're right.
Okay, I'll let other people ask questions.
Yeah, I can hear you now. Yeah. Is it only me, or can others hear Sonia?
I can hear her. I think there's a lag in somewhere.
Okay, I think I lost everybody now, maybe.
You haven't, Bhavna. I think there's a lag at your end because I just heard Auntie.
Okay.
Oh, but I think it's okay now. You're seem to be...
Now I'm
okay.
Fine. I'll just stop my video. I think perhaps that's adding to the thing. Yeah.
Okay, do people have there are 9 minutes for questions, so do bug Bhavna. It's nice that we can take it slowly. So I wonder if anyone has noticed any parallels with the vertebrate stuff that we looked at earlier. Invertebrate stuff that we looked at earlier.
Yeah. Yes.
So then I'll ask another question.
Yes. So was your previous question on whether these are regionalizing factors?
Yeah,
the answer to that is yes. Yeah.
You answer. Yeah, you answered that. So then LHX2, Bhavna, is acting as a regionalizing factor, but it also seems to be having a role as a temporal factor. So it's kind
of both.
It has these dual roles.
Yeah.
And then it's interesting that so are you able to tease apart in the phenotypes that you have whether it's an early role of LHX2 that is giving you those time specific the inhibition of layer 5, or whether it's the late expression that's giving you that domain, that identity change?
Yeah. So the answer to this question is that, so I think across cortical development is mostly there is an integration of both spatial and temporal at the same time, right? So spatial is purely that where you are in that gradient across the rostrocaudal or the dorsoventral axis, and temporal is the time, which there is like an internal clockwork which is happening. And some of these spatial cues are all driven at the very start of the patterning itself. For instance, FGF8 has already driven the entire cortex into a region of, anything close to the FGF8 will become motor cortex. Anything farther away will become the visual cortex. So it's already driven. So once that drives and it makes these, gradient expressions, there the spatial has already started. Simultaneously, all the temporal has also started. So at any point, it's both integration of both spatial and temporal, not one versus the other. And I'm sure many of these, just like LHX2, though it's not been studied, would be doing both, depending on whether they have a gradient of expression and all of that. Now, coming to the question of whether it is an early role of LHX2, the Cree line which we've used, actually it's an early driver line. It's around 11.5. Yeah. So we don't have currently, but a later driving Cree to look at whether, LHX2. Now, interestingly, if you see here, that LHX2 is not just expressed in the VZ progenitors, but also it's coming up in the mature post-mitotic neurons, right? So we would like to have that very specific Cree where we can remove only, because the moment you remove LHX2 from here, your all early defects come up. So you cannot now see what role it's having in the post-mitotic neuron. Yeah. Similarly, LHX2 is still being expressed when your upper layers are being generated. So that's something which you would like to ask and answer, that, how perhaps, one transcription factor and maybe many transcription factors, they drive this entire process temporally. And one thing which regulates this temporally is just one study, and perhaps a clue is that the chromatin is actually closing down as the neurogenesis proceeds. So the chromatin which was more open during proliferation keeps closing. So which means that there is this restriction already happening, which starts from the very first division itself, that once it divides, the progenitor divides, its chromatin starts closing. So that's one way to look at how this temporal thing may happen during development. Yeah, so I think the thing is we need to have more crease. But now I think with single cell RNA-seq and people doing it across different axes, that will perhaps just change the scene completely. So I'll in my next talk, I think I'll start where I'll just probably give you a glimpse of this single cell RNA-seq study, which is from Denis Jobadon's lab, where he gives very beautifully, talks about how the temporal patterning happens, that how the architecture of this progenitor itself is changed from an introverted to an extroverted cortex. Like you can see here that the expression of genes go from chromatin organization to membrane receptors in these progenitors, making it more receptive to external cues. And so this is one axis which is across the VZ, but the other axis, which is like as it go from progenitor to the neurons, there also the expression changes, making it more neuronal. So I think this will probably give more idea about this temporal patterning.
Cool. Yeah, that sounds exciting.
Yes.
So do people have any more questions?
So I have about 12 slides. I'll probably take half an hour and maybe say another 10 minutes. So a 45-minute class should do. So maybe I should just, you're getting to Raj? Okay.
Yeah, I just wrote to Raj and I copied Varsha on it. And so we'll update people ASAP. Does that sound okay, everyone?
Is that okay with everybody?
Yes.
Yes. And just to give you a preview that, I'll take you now through organoids and some of these human brain development. So that's what is coming.
Yeah.
Just to keep you guys excited.
Yeah. Okay.
Can I ask one question before?
Yes.
Yes. Sorry, Yojit. Yeah, go ahead.
Please.
Bhavna, you just mentioned about this chromatin accessibility, inaccessibility phases.
Yeah.
So I'm just wondering, why does the CNS give rise to neurons first rather than giving rise to the glial cells? Because the chromatin, so I think after this chromatin is inaccessible in neuronal stage, but it's accessible when the astrocytes start to form, right? So why not give rise to astrocyte and then give rise to neuron? Is the question?
So the answer to this, I'll probably modify the question a bit, and there are elements of it which I would like, which are very, very interesting and very important to ask. So it's a good question. So one correction is that the chromatin doesn't become inaccessible. It's just that it becomes restrictive. For instance, you have more open chromatin to less open chromatin as development proceeds. But having said that, I must say that what really happens in glia production is that several of these glial, so there are two ways to make glia, right? One is that you, one view is that you have certain, you have progliogenic factors even during neuronal development. So what is happening is some of these neurogenic cues, for instance, LHX2 also. So the progliogenic cues are Notch and NFIA and all of that. These cues are even present during neurogenesis. So one way the glia are stopped from being produced is that these neurogenic cues, they repress the expression of all these gliogenic molecules, right? So, and as time progresses, because the chromatin accessibility changes, some of these neurogenic loci are shut off, and that's how the repression is gone and the glia are being produced. And think of it in the same way as I talked to you about the switch, layer 6 to layer 5 switch, right? So during layer 6 production, you are keeping the layer 5 production switched off, and then that repression is relieved at the time of layer 5 production, which in turn goes and represses. So that's something similar which is happening from the switch to the neuron to the glia also. Another thing also happening is once these repression is relieved, these gliogenic molecules go on and activate further very more important glial genes like NFIA. The notch itself goes and activates NFIA, which is the moment you express NFIA, the entire cortex becomes glia-like, right? So there are two ways to do it. One, you remove LHX2, which is repressing the glio production. You would still, if you remove LHX2, you would still get glia, or you overexpress NFIA, which is a progliogenic molecule, and you still get glia. So that is how the switch is happening. And coming to your question of why glia didn't come to neuron, I think, so, I mean, we don't know exactly. You can, of course, remove LHX2 and you would actually see precocious gliogenesis. You overexpress NFIA, you would actually see precocious gliogenesis. So it is possible. It's just that later on, they don't go on to form neurons. But I think one of the reasons why this may happen, and this is just what is thought to be in the field, I don't have a concrete answer to it, is the number of glia outnumber the number of neurons. So all these glia which are produced in the cortex, they match the numbers of the neurons which are produced. So I think one of the reasons why the glia come later is once you have the basic skeleton of the neurons, you want the number of glia start producing so that, you know, you match to those numbers which are produced. And I think in the human cortex, we have about 83 billion neurons, but 1 trillion glia. So that perhaps could be one of the reasons, but I mean, I don't know evolutionarily why this, because in Drosophila, they both produced at the same time. Now, I really don't know why it switched and it became a temporal thing in mammals. Maybe there is some study out there. I think we can look it up, that why it could have been this switch from producing both neuron and glia at the same time to a temporal timing happened. I think one of the reasons is to match the number of neurons which are being produced so that the glial numbers can be matched to that. Does that answer your question?
Yes, yes. Thank you.
Yeah.
Okay, I think we should let people leave now. Thank you so much, Bhavna.
Yeah, thank you so much, everybody. I hope
we'll
you,
let
like
you
the
know
class.
as soon as we know about the next